Okay, thank you. Good afternoon, everybody. And well, first of all, I will thank Rowan for inviting us to speak about these food safety issues, bacteria. So the speech will speak about the VTEC, or which is also called cheesemaking, Escherichia coli, which is also called STEC. And I will first of all have a bit of description of what are VTEC, with a bit of definition. Sorry, but I need to do this for you to better understand what are these bacteria. Then I will give you a few data about the fact how they can affect health and could be a food safety issues as well. And then give you a bit of explanation of the way we have to analyze them and to see if they are present or not in cheeses and in raw milk products as well. Then give you a bit of explanation of what do we know, and I should say what about we don't know about their behavior in cheeses. And then try to give you a few tips in case if you've got a positive sample for VTEC, and then try to have an overview, a really quick overview of scientific data and works that are in progress about VTEC. So, beginning with VTEC definition, well, when we say VTEC or STEC, we said Escherichia coli. So Escherichia coli is just a normal bacteria, usual bacteria that you find in the gut flora of human and many animals. Usually most of them are harmless, and hopefully you and I are living quite well with all these bacteria, but some of them can be pathogenic, and they could lead to different symptoms coming from diarrhea to bloody diarrhea and to urinary infection towards more severe symptoms, which is called the hemolytic and uremic symptoms. And in fact, in the big family of E. coli, you've got different kinds of bacteria having different kind of genes. And first of all, you've got some of them that have got the STX genes, which is the cheesemaking genes. And then when we've got an E. coli that are having these genes, you are speaking about STEC. But among them, you've got some of them that are having other genes. And then you're going towards different kind of bacteria that are more and more dangerous ones. And we consider some of them highly pathogenic, or consider them as the most pathogenic STEC, and they belong to a different serotype, and they've got a few amounts of them, but I will just explain that a little bit further. So the VTEC is not all the E. coli, but really a particular subset of E. coli. And we're speaking about EHEC when they are isolated from humans, so from people that have been healed. And you've got different symptoms, as I've already said, like bloody diarrhea, towards hemorrhagic colitis and HUS. And unfortunately, they're quite famous all over the world because they've lead to a lot of food safety issues and with severe complication as well. And we can consider that it's emerging pathogens with a really high amount of cases all around the world. So, as I already said, they've got particular genetic features. The first one, is it that one? Yes. Does it work? Yes, I've got it. The first one is the fact that they are able to attach to your flora near your gastrointestinal tract, and being there, they're trying to, and they begin to produce the cheesemaking, which is the toxin that leads to the bloody symptoms, with your diarrhea becoming bloody as well. And the fact there is also different pathogenic factors that I won't give you in details because there are a lot of different genes and different, well, let's say abilities they've got, but they are able to get rid of the immunological reaction of human and get rid of that. And the things that you should know as well is that what cause the disease is the living bacterium. It's not the cheesemaking that has been preformed in the food that make you ill. It's that when you ingest living bacteria. And what we know is that we need a really low amount of bacteria, up to 5 to 10 bacteria that could lead to disease, especially when you were speaking about cats. So, when we are speaking about VTEC, the problem to recognize them and to separate them from the whole range of E. coli is that they've got no particular physiological properties that will facilitate the distinction in between E. coli bacteria and VTEC bacteria. There's a little bit of exception for E. coli 057. We've got a particular sugar metabolism, but otherwise the other VTEC are quite not different on a physiological point of view from the other E. coli. So just to make sure of what we are speaking when we are speaking about VTEC, and of course we can't fight against all the family of E. coli. We've got set up classification based on the serotypes that have been most often involved with food safety issues. And it's why we have been able, or at least the authority of different countries, have defined the most pathogenic serotypes of VTEC. And fortunately for us, it only concerns a few serotypes. So the serotypes, what is it that? It's just, let's say, the outer membrane of the bacteria and one of the flagella, which is called, what could be considered as a tail of the bacteria, which got different antigenic structure, and it could differentiate different E. coli. And we've got nearly 200 different serotypes, considering the O difference and the H difference. But hopefully we're not fighting all over 200 different serotypes of E. coli, or that's just the good point, is that when they do some risk assessment and analysis based on food safety issues and human data, Europe with the European Food Safety Agency, France, or even USA with the FDA, just distinguish 5 to 7 different serotypes like being the most pathogenic ones. And just to explain that, in fact, you've got example here in Europe when they take up and they consider data of E. coli foodborne outbreaks during 5 years nearly between 2007 and 2011. And they look and they characterize the E. coli serotype, and what you can see is that for most of the case, most of them were linked to 0157, 026, 0103, which leads them to give top 5 or more serotypes E. coli for VTEC. And when they're looking for their genetic features, they see that looking of what are the genes and the particular subset of genes they've got that have been involved in the food safety issues, let's say considering the presence of the genes coding for the cheesemaking or the Vero toxin, we've got VTX1 and VTX2 because you've got 2 different variants of these cheesemaking genes. And for the genes that is responsible for the fact that the bacteria attach to your gastrointestinal tract, let's say that nearly 80% of the cases were linked with the presence of these genes. So that leads, of course, to give an explanation and a characterization of this bacteria. The second point is, as I told you, well, you've got huge different amount of and different kinds of E. coli in front of you, and we need to characterize 5, or let's say 5 to 7 different serotypes of E. coli. So there is not an easy way to analyze them, and most of the time we've got another drawback is that we are looking for a very low amount of harmful bacteria. So I told you 5 to 10 bacteria in the food that you ingest can be harmful. So the first thing that we have to do, of course, is just sample your food, but then we proceed towards an enrichment step, and then we detect the bacteria, and then we are sure that it's there. So there's a few points to take care. As we are looking for a very low amount of bacteria, we usually take care of having a 25 grams sampling of food. And it's quite important because we've got low amount of bacteria and we usually have got scattered contamination. So we need to take care of having enough sample. Then the second step is just to proceed to the enrichment, which is quite, well, let's say the tricky step, because we use selective broth and we're using different antibiotics, and it's just to be able to have more coliform growing than other bacteria. Then we're doing toward the detection step, and this one is that you've got your enrichment broth, you're extracting the DNA, and you're looking for the genes that are indicative of the different serotypes we speak about. And at this stage, you just only have a presentive results, which means that you are looking for genes, but it's a bit like if you were looking for bones. You're not sure that you've got a living bacteria anyway, you've got just genes, and it's the genes that will make you sick. So the next and the final step will to proceed towards what we call a confirmation, and then you are going towards isolating colony. So you're taking your sample back, and then you're looking if you're able to cultivate a bacterium, and then, which means that you've got a live E. coli, but the other point is that just to be sure that E. coli get all the genes that make you sick, which means that you've got the genes coding for the cheesemaking, the genes that is the intimin, which is the genes that make the bacteria attached to your gastrointestinal, I won't say it, flora. And then, of course, what is the serotype, the O157 or the O26, for example. So this is quite a very long step. And on the left, you can see that, well, at the first day, day 0, you're having your food, then you're going to a PCR testing, and then hopefully by the end of day 1, you've got a presumptive suspicion, and then you're going back again, and you're trying to isolate your living colony, and then so by the end, you will have your final results only by day 3. Hopefully you don't have a weekend step at the middle of that. So it's just a long and complex method, which means that you don't have the result as quickly as you want, or you usually want. And the other thing is that there's a lot of steps with PCR testing, etc., which means that it's quite an expensive method as well. What do we know about VTEC in cheese is, in France, we've got the chance, I don't know if it's really a chance anyway, of having a survey that have been done by our food administration, and there have been 2 annual monitoring plans conducted in France, and they were based on the isolation of E. coli, having the genetic features showing that they would be highly pathogenic E. coli, so which is the presence of the gene coding for the chigatoxin, the intimin, as well belonging to the 5 most pathogenic serotypes I told you before. So a few years ago, nearly 2,000 raw milk cheeses were analyzed, and we found a contamination rate that was 0.9%. 2 years ago, the same monitoring plan was done as well, with a little bit less cheeses, because there were nearly 1,000 raw milk cheese that were collected, and we found it a contamination rate of 0.2%. So which could be quite a bit low anyway, a low incidence. But speaking about the serotype that were identified in cheese, most of them were having the serotype 026, which was the most frequently isolated from cheeses. So in conclusion, well, we are a bit happy with the results, saying that we've got a low incidence, but it's a low incidence because we are considering the fact of having living bacterium in the cheese with all the genetic features I told you. If at one stage, if we would have been interested in, for example, just looking if cheese, if we could find the cheesetoxin genes, we could nearly have 1/3 of the cheeses that would have been positive. So it was quite important to go towards and just to select what are supposed to be the most pathogenic bacteria, just to be sure that, well, we are looking at the top of the iceberg anyway. Then, what do we know about, I told you we don't have a lot of, the incidence of VTEC seems to be quite low, but cheesemakers and especially a raw cheese milk maker were interested in understanding their behavior in cheeses. So a product program, a research program called Foron that have been supported by the French Dairy Federation, the CNIAL, have been conducted just to see how steak were growing and survive in different kinds of cheeses. So I will just to show you a few results there. If you've got results done with challenge tests with raw milk cheeses, and basically we inoculated raw milk cheeses with nearly, well, between 50 to 100 steak per milliliter, and then look how they were behaving. So here you've got their behavior in semi-soft cheeses, and you've got the first 24 hours of cheese making, and here you've got what happens after 40 days, so during the ripening stage. And we've got results that see that, of course, they will develop really early in the cheese, in the early stage, and at 5 to 6, 7 hours, they will be at the highest level. We try to observe if there were any difference in between the different serotypes and the most pathogenic one. There weren't a lot of difference except for the red lines you've got there, which are the 0157 serotype that seems to be lower than the other ones. Just another example is the way they behave in soft cheese, which is more like a camembert type cheese. So you've got the first days, and then we are going over 40 days of ripening. And you've got different serotypes in different colors, and the same thing. You've got a really huge amount of development increase during the first hours, and then most of them survive during ripening, except for the green one, which is 0157, which tends to be decreased. And there was a bit of difference in between the rind part of the cheese and the core part, with the amount of VTEC being a bit higher in the rind part of the cheese. So, just to conclude about their behavior in cheese, because they were tested on different kind of technology, we've got a growth for the different strains inoculated, in particular for 025, 01, 03 and 145, and the growth was not so high for 0157, sorry. The maximum increase is observed during really the first day and the first 24 hours of cheese making for soft and semi-soft cheeses. And then there's a bit of difference between these soft cheeses and semi-soft cheeses. They stay in the same concentration all over the 40 days of ripening for semi-soft cheeses, but trying to be a bit of decrease, a little decrease for soft cheeses as well. And there is a decrease that was observed in hard cheeses, because we've got a cooking step at 53 degrees, so it's probably at least the high temperature that will limit their growth, and they won't grow as well in lactic acid cheeses, probably due to the presence of lactic acid that could inhibit them. Well, knowing that, try to go to a more technical point of view or a more practical point of view. What to do if a cheese sample is positive for VTEC. So what I will explain is that what we are applying in France, considering that our French authority usually look after the 5 most pathogenic serotypes, and they are not, they consider that they are the more dangerous, so they applied legal things and prevent any cheese being on the market for these 5 most pathogenic serotypes. So for example, you've got, the first thing is that you can find having a result coming from a supermarket control and saying that, well, we've got presence of VTEC positive. So the first point usually we ask is what the exact result we've got. What are we speaking about? Are we speaking about enrichment growth results? So if that were just only on a presumptive presence, we're speaking about genes. As I told you a little bit further, we're speaking about balls and not living bacteria that could be harmful. So the first point is then you should ask for colony isolation, or on the other way, you've got isolated colony results. But anyway, if you've got isolated colony results, you should ask for a bit more details about the VTEC characterization in, let's say, what kind of cheesemaking genes you've got, and of course, what are the serotypes you are looking after. Well, anyway, by no chance, you've got, for example, a cheese batch having 0 to 6 serotypes and having cheesemaking 1 genes and antimine genes. Well, first of all, if you've got the MPS tag detected, we are not allowed to put the batch on the market, so if it's already there, you have to withdraw it. And a good thing is, of course, to look after the other batches, so the batches that have been performed and made the day before or the day after, and just to look, sampling them and check for their presence of VTEC. And of course, usually when we are looking for low amount of contamination, scattered contamination, they recommend to do 5 samples and 5 different samples, and each sample being a 25-gram sample as well. And the objective for the food factory is just to be sure if you've got an isolated problem or not. Then there are a few tips that you can do, is that, of course, just to be sure that at the cheese process, going through the GHP and SSOP practices, just to be sure that you, while focusing on E. coli or any fecal routes, and maybe reinforce cheese control as well. We could ask the interest of having analysis of raw milk that coming through the cheese factory, but well, it would not be so easy because, well, you have to sample and analyze is raw milk from farms, but remember that we are looking for a really, really low number of bacteria, and they're really quite diluted in milk, so it's not so easy. Well, there are a few tips. For example, some laboratories can go toward clotting of the milk just to, well, make a little bit of concentration of the milk, while get off with the water and having a bit more quantity of milk to analyze. And at the farm level, the objective is to limit fecal contamination of the milk, but then I will, let's say, well, Sabrina will speak a little bit further about that, so I won't say too much. And that's usually the minimum to do, but I should say that a few cheesemakers go towards problems of VTEC in France, having, well, a serious problem as well, and it's not so easy to get rid of, because the really tricky point is that we are really looking for small amount of bacteria in milk, and we are not usually easily found, which is the milk producers that could contaminate the milk, or, well, it's a fecal contamination, so we shouldn't. It could be also an employee that could be a carrier of E. coli as well. Then I will finish by giving you a bit of scientific data on VTEC and a few things that work that have been done of E. coli. So that's a research program that are currently performed in France. And we think that the point is that we don't have a lot of food safety issues linked to VTEC coming from raw milk cheese. So we think that our dairy strain, or VTEC dairy strains, got particular features. And when we analyze the strains that have been coming from dairy origins, which are most of them being the 026 or 103 serotypes compared to human or environmental strains, they belong to different genetic clusters. So, well, there's probably a pathogenetic significance, but we are not able yet to explain it. And what we were able to show is that these bacteria have got a low ability to disseminate the gene coding for the cheesetoxin, which is a good point, because at least this dairy strain are not disseminating the cheesetoxin pathogenicity factors to other bacteria as well. And when we characterize the E. coli VTEC isolated from cheeses, most of them have got the STX1 variant, and which is a good thing because it's really less harmful than the STX2 variant. It's more like 1,000 times less pathogenic, and probably because it's got different way it will be put in the genetic chromosome of E. coli. But the other thing is just considering as well the raw milk cheese matrix. Of course, we are speaking about raw milk cheese, and there is competition with endogenous microflora, and so it could contribute to lower VTEC growth. And I think that, well, Valérie could say a little bit more about the role of AFNIA limiting the growth of VTEC. In cheese, there is also a lot of organic acid production, especially the lactic acid production, and we know that it could decrease VTEC during ripening as well. And what is quite interesting as well is that, well, we've got good creamy cheeses, which good fat, and the fat structure just binds to E. coli receptors, and it prevents them from attaching toward the gut, which is a way of preventing or limiting food safety issues for these VTECs. But this is a few tips, and there is still a long way towards better knowledge of dairy VTEC strain, and there's still a lot of questions concerning VTEC control at farm level. So there's still a lot to do, even if there is a lot of research programs. So just to conclude, and then I will let Sabrina talk, if you just remember a few things, that when we are speaking about E. coli, we're just speaking about a small amount of E. coli in this big E. coli family, and they've got specific genetic features that unfortunately lead to serious health problems and complications. We need a low dose of ingested bacteria to make you ill, as long as they are alive, which is quite an important point. And unfortunately, they are not enough specific to distinguish them analytically in an easy way. In cheeses, they easily grow in soft and semi-soft cheeses. They're usually present in low amount in cheeses, which means that when you need to sample your batch, you need at least to take 25 grams sample, and maybe having 5 different samples as well. But our data, we've got differential that we've got low prevalence in raw milk cheese, less than 1%, And if you've got one cheese positive, then, well, be sure what you are looking for when you are speaking about VTEC. Check the other batches. Be sure of your GHP SOP practices in the cheese processes plant, and at farm level, at least you need to reinforce hygiene milking practices. But that's a few tips, and we are sure that there's still a lot to learn about VTEC and the way to manage them. well, thank you very much.